So I'm going to talk about using eDNA. I start off with fish and then I move on from there. And there are a ton of people working. I actually have multiple projects that intersect with eDNA, and I'm just going to talk about a little of them. And I stole some of Chris Peters' slides, so I'm going to put them in as a co-op. So environmental DNA, how many people here are working with or have a project that is incorporating environmental DNA? That is not as many as I would have thought. Okay, so environmental DNA is just DNA that's extracted from an environmental sample, be it soil, be it air. In most cases, it's water for a variety of reasons. The concept is not new. There are people in this room who have been doing this type of work for a long time. We don't need to talk about how long, perhaps. But what is new is the cost and the availability. So the cost of doing these analyses is rapidly dropping. The availability is increasing. So most universities can do this. There are commercial labs that can do this. So we now have the access to this technology that we really haven't had. And what we're trying to do is figure out, how do we use it? How do we make it useful? What can it give us that we didn't have before? So I'm just going to walk through a quick example of some work we're doing in Great Bay. We are doing some work in coordination with the Givner Swamp Program, where when they collect the swamp samples, they also collect eDNA samples. So if we do a comparison between the fish that we detect using eDNA, and those are the blue sites, versus the same sampling that Connor just talked about, and those are the yellow sites. These are not all of the sites, but these are the ones that are sort of close together, so it's good for a comparison. This is data from 2022. The sites are not exactly the same, the dates are not exactly the same, but the scale of the effort is fairly similar. And if we look at the fish species that we detect, so the seining, just for those sites and those dates, there are about 25 species. For the eDNA, there are about 39 fish species. There's overlap of about 18 species. If you look at the species in the seining that are not in the eDNA, they tend to be either species that are more likely to be out of the mouth of the estuary where those sample sites are, or places where there were just one or two fish and we didn't do the eDNA sampling at the same time. The eDNA species, the reason we have a lot more species in the eDNA is a lot of those are actually freshwater species. So we're sampling at the bottom, near the head of tide dams, water's coming down over that dam and is carrying DNA from freshwater species. So that's either a minus because we're potentially getting erroneous detections, or it's a plus because we're getting a 2-for-1 because we're actually sampling up the stream as well. So when we look at those two different methods, we're fairly confident that we can use eDNA at this scale, at this level, to reasonably accurately monitor just the general consortium of fish that are here. So we can say, okay, we can use eDNA for fish monitoring in Great Bay, but you're already doing fish monitoring, right? So I just went to a whole lot of work to tell you that you have mummy checks in Great Bay. You don't really care. Why is it useful? So here are the two places where we're going from here. One is that we are actually part of a national program. So we're sampling at 10 near sites around the country. Not all of them do fish monitoring. So we're validating the method here. And several of the other sites, we're doing these comparisons. And that gives us the confidence to say, yes, we can use this method in estuarine locations. The other, and then also in areas of Great Bay where you're not doing monitoring, if you want to be able to use eDNA for fish monitoring, that's a potential use. We've done a lot of this in freshwater systems, so streams, above and below dams, those kinds of places. So make sure Chris isn't standing up it. And then that's the rest of the near sites. But the other thing that is really potentially useful is the sort of the untapped DNA in these samples. We take a sample, we extract the DNA, we're not just taking out the fish DNA. We take a very, very small amount of it and we run an analysis and the rest of it goes into a freezer. We can pull that back out. So we pulled some samples and Fuad, who is sitting right there, does this actual analysis for me. We ran a couple of the swamp samples, there's just a few samples for birds. We ran it for mammals just to sort of see what we would get. There's nothing deeply surprising here. They're all things that we would expect to see, but that's another use from that same sample that we already have. Possibly even more interesting is we can run these samples for very broad, say, eukaryote species. If we take the 2022 Great Bay samples, there are over 1,000 genetically different things in those samples. We don't necessarily know what they are by name, but we can tell roughly where they are in the phylogeny. Some of them we can name. And we can potentially use that to tell us how the system is changing, or if there are particular characteristics in different areas of the system. So if we look at a per site, this is just one set of samples. These are 4 of the Great Bay swamp stations. There are very different communities. These are the copepods, these are the algae, these are bits of fish. A lot of it is microscopic organisms. They're very different at different sites. If we look at it over time, it changes with time. So, and that's not surprising, right? You expect to have different things in the water in June than you do in October. But we can use this method to start to say what is changing. We can go back into these analyses and say, these are the particular species that shift with time. We can look at whether or not certain species shift in response to some pressures. What happens when we have a very dry year? What happens when we have a hot year? We can take a look at what's changing and think about how that is potentially trickling down into the whole ecosystem. And so my sort of question to you is, I'm not the biologist, I'm just out there collecting this data. We should be thinking about ways in which this untapped information is potentially useful for us to help understand and manage Great Bay. Thank you. That's because I gave you a question, so that way I wanted you to have lots of questions. I get to pick them?
Yep.
Okay, Celia?
Thanks, Alison. Yeah, I've been really wondering about the DNA. It's great to hear it's more available. I guess I wondered what you thought the application of would be for long term. Like, we're all worried about changes over the long term. You think that is the variability of things too much that you could say, take samples in the spring every 5 years? Do you think that change would be meaningful? Even if you didn't know what this was called.
So I think so, but I'm gonna have to fall back on I'm not an ecologist, but over the long term, I mean, if ecosystems change, if things heat up, salinity changes. I mean, we know that things like copepods as indicator species shift. And all we're doing here is we have a new, we have a more detailed way of measuring things that we kind of already know are there, but we can get more information about them in more detail. So we do have samples from Great Bay from 2018. We have not done that time series, but we are starting to gather enough information to start to look at that. All right, we'll go to Steve.
Yeah, Alison, when you're talking about your 1,000 species of eukaryotes that you can get at, when you take, I was looking at the profiles, and it's kind of interesting what's popping up in some of them, but that's going to reflect what's coming over the dams as well, and from the land, right? So that's another complication there.
Yes, so potentially, particularly in this sampling regime, because we're sampling some of this footage time.
Yeah.
Interestingly, in the site that's in the middle of Great Bay, we do not see the freshwater species. So that's an indicator to us that we don't, it doesn't transport fully through the bay. Yeah. Easton?
Yeah, Easton White UNH. I think from my biologists had on. I think the thing that I'd be most interested in going for, I know like kind of the ways for all the relative and absolute abundance and getting at that much as opposed to just species. So really what you're allowed to thinking about that aspect.
Yeah, so that's work that is great. One of the ways we can get at it is through replicate samples. So some of it's just the more times you see a species in a sample, the more likely it is that there's a lot of it. And then there is work calibrating to abundance either through genetically or by basically calibrating with a known abundance. And in streams or places where things are perhaps a little less complicated, people are really getting there. I would say in estuaries we're still a ways off on that. Yeah, okay, we have one there, and then are we running out?
2 more questions.
Okay, 2 more questions.
That wasn't.
That was your question, okay. Um,
Hi, Jenny from UNH. Just going off of what Celia was asking in terms of directly to the future, wondering if your group has thought about any historical reconstructions of eDNA using like a paleontological approach, something like that, informed from different areas and into the Great Bay.
yeah, so people do that. I have not done that. There's a lot of details involved in doing sediment cores and turbated systems. But yes, people are doing that and getting super interesting results. And I will just leave you with this one thought. This is actually from Japan. These are a whole bunch of samples they just took around the country, and for the heck of it, they threw it out against anthropogenic stressors and found that there was a very clear distinction between the community of eukaryotes and different land use stressors. And that's the sort of thing that we hope to be able to get at with our multi-years countrywide project. All right. One more for Cory, because she's
Yeah.
special.
Well, also because it's so related to that, I was just curious, if you think that, that interesting graph of the changes in, types of eDNA that you're finding, like, could you look before a wastewater treatment plant went in
Yeah.
and after?
Yeah.
And
Yeah.
have you done that yet?
No. But then we have to put a wastewater treatment plant in.
But, well, 2018, I don't know, like,
So we could look at the after, right?
you need before and after.
We need before and after, but generally, anything, you think of all the ways in which we measure change, so the copepods, the benthic invertebrates. This is just another finer comb for
Yeah.
getting at those same things.
Before, after.
Yeah. Okay. All right.